Hello everyone. My name is Gaurav Bhalerao. I'm postdoctoral researcher at Department of Psychiatry, University of Oxford. I'm going to talk about my work on automated QC of T1w scans for clinical datasets, methods comparison, and design of a quality prediction classifier. Before my talk, I would like to mention that I have no disclosures. In my postdoctoral fellowship, I'm working on Dementias Platform UK project. DPUK brings together scientists, researchers, and clinicians across the world for finding new ways to detect, treat, and prevent dementia. DPUK also provides a data portal and analysis platform to fast access and analyze data. DPUK has 40 cohorts of dementia, of which 19 cohorts contain brain MRI data. And in this project, I am working on adapting UK biobank imaging pipelines for DPUK data. T1 weighted brain MRI is a key modality in neuroimaging analysis. It is essential for registration, also for estimating brain morphometry. And the quality check of T1 weighted brain MRI is extremely important for ensuring reliability of outcome measures. So here is a typical example of good quality and bad quality T1 weighted MRI. In terms of clinical data sets, QCing T1 weighted brain MRI is often challenging because of the artifacts, motion, and higher atrophy due to aging and neurodegeneration in clinical populations. Also, manual QC is time consuming and subjective. Automated QC tools are developed, but they are almost always developed on healthy young subjects' data. So, in this work, we would like to develop a QC framework for clinical data sets from elderly population that is generalizable for multiple sites. So objectives are: first, implement automated QC tools on clinical data sets, relate and compare the QC metrics and ratings from these data sets, and then design a classification framework for quality checking large clinical data sets. So this is the flow of our methodology. We collect T1 weighted brain MRI from clinical data sets. We get the visual QC measures from cohort owners, and then we process these images in MRI QC and Cat12 segmentation pipelines. After we process these images into automated pipelines, we compare the quality measures in correlation, and the ratings from these two tools and visual QC are compared with each other using inter-rater reliability. And finally, we design our own classifier by combining the quantitative quality measures from both automated tools. Let's have a look at the datasets that we used in this study. We used more than 2,000 T1 weighted brain MRI scans from patients and controls from 3 scanners, 3 field strengths, and 39 scanner manufacturing models. So have a look at this table to see more details on the datasets. Our datasets contain age range from 55 to 116 years, and these datasets contain heterogeneous acquisition protocols. First, we obtain visual QC from dataset owners, and each dataset has different categories of visual QC. So after a careful inspection of the description of these categories, we bin the images into accept and reject labels. Then we process each anonymized deface image into automated tools. So one of the tools is MRI QC. We obtain 68 quality metrics, and then we use MRI QC classifier to bin the images into accept and reject quality. Whereas in the other tool is computational anatomy toolbox. So it provides image quality ratings of about 36 image quality ratings in the segmentation pipeline. And then we use weighted image quality rating to bin the images into accept and reject quality. So just have a look at these reports that automated tools create for each individual image. After this, we compare the quantitative quality measures created from these automated tools using correlation analysis. And what we found was several pairs of measures had strong significant correlation. However, there are some measures which showed poor correlation, and these can be the unique measures from both of these automated tools. In the next step, we compare the accept reject counts from all three methods, manual or visual QC, MRI QC, and CAT12. What we found was CAT12 had higher acceptance percentage as compared to manual and MRI QC, whereas manual QC has more acceptance as compared to MRI QC. So in the next step, we compare the ratings using the inter-rater reliability analysis. So we calculate the agreement between these different methods. And this table just shows what are the default ratings from the automated tools. And for visual QC, we have already binned the images into accept and reject labels depending upon the categories. So what we saw was CAT12 and MRI QC, CAT12 and manual QC has higher agreement for Whitehall 2 dataset and the least agreement for OPDC dataset. Similarly, uh, similar was the case for other comparisons. But one thing to note is the agreement varies for each dataset. So it was very difficult to make any judgment on which tool produces uh, produces the ratings which are closer to visual QC. Then we change the ratings of the automated tools to strict and lenient thresholds, and then we recalculate the inter-rater reliability values. And what we again observe is there is improvement in these ratings. There is improvement in the agreement, but it again varies for each data set. So for some data sets, we see the improvement, but for some data set, we do not see any improvement. So it is again very difficult, even after changing the thresholds, to have a judgment on which automated tool produces ratings closer to the visual QC. Hence, in the next step, we design our own classifier by combining MRI QC and CAT12 QC measures. So these are total 104 measures, and we use visual QC as the target. And in the classifier design, we use random forest classifier in nested cross validation to optimize the model hyperparameters. And these models were designed for different feature sizes. So two primary models were designed, of which the combined data model is the combination of all the data, combination of all the data or samples from all data sets. And here we divide the data into training and testing randomly, but then we try to maintain equal proportion of each site in this combined data model. In the next, we validate this combined data model using one site out model. So in the one site out model, we leave each site as test data and train the random forest classifier on the remaining data. So let's have a look at the classification performance. What we found was combined data model outperformed CAT12 and MRI-QC ratings. Also, average performance of one site out models was significantly higher than CAT12 and MRI-QC. So you can also see the specificity of our models is quite higher than CAT12 and MRI-QC. So we observed what are the top 10 ranked features from both these tools, and we found that summary measures from white matter signal to noise ratio, summary measures from background in MRI-QC were ranked at the top. Also for CAT12, these were noise measures, surface measures, and then gray matter, white matter, background measures were ranked at the top. So we had a look at these top 10 ranked features, and we just plotted them across our data sets. And what we found was actually these QC measures can distinguish these data sets from each other. So that proves the technical and other sources of variability between the sites, and these QC measures are wonderfully capturing this variability. So this forms the data harmonization work that we are planning in the future. So in summary, automated QC tools provide similar QC measures, but direct implementation of these measures is challenging. Inter-rater reliability of ratings from automated tools with visual QC depends upon the acceptance threshold and varies for each dataset. Our classifiers outperformed existing QC tools and appears promising for heterogeneous datasets from multiple sites. Combining quality measures from both the tools contributed to wider coverage over different datasets, age range, and scanners. So in future, we aim to utilize this framework for improving generalizability of prediction, and we will release these classifiers on the DPUK data portal to robustly QC T1 weighted scans for other DPUK cohorts. So I would like to thank all my co-authors, my supervisors. I would like to thank DPUK and ADNI for providing me data access. And finally, I would like to thank OHBM for providing me with this opportunity. Thank you for listening.